Welcome to True Projects. In this video, we will be explaining about phylogenetic tree construction based on biological datasets of cyclooxygenase, which is COX1 and COX2, by using cluster analysis based on experimental values. Introduction. Fast improvement of high throughput technology in biological research has resulted in massive volume of genetic data gathering. The abundance of genetic information which is now available has changed biological research, providing new insights into relation between different species and their evolutionary histories. Phylogenetic trees are important in illustrating the evolutionary relationships between biological organisms and genera based on similarities or differences in their physical or inherited features. Traditionally, development of phylogenetic trees has centered on examining significant organismal traits. However, with the arrival of large-scale genomic information, researchers can now investigate genetic similarities and differences at the molecular level. We use gene dataset in this project to study similarities of genes from different species utilizing cluster analysis approach such as hierarchical clustering, k-means, and self-organizing map, which is SOM clustering, with principal component analysis, which is PCA. Objective. The goal of this project is to evaluate the genetic dataset of cyclooxygenase, which is COX1 and COX2 genes from diverse species using cluster analysis approaches such as hierarchical clustering, k-means, and self-organizing map, which is SOM clustering with PCA. The project goal is to build a phylogenetic trees that reflect on evolutionary relationships between species by discovering similarities and trends in gene behavior. The idea is to graphically highlight the commonalities of grouping genes with similar patterns within the same branches of the tree. These are the requirements for execution of the project. In hardware requirements, operating system of Windows, processor of i5 and above, RAM of 4 GB and above, hard disk of 20 GB and above are required. In software requirements, Python IDE of 3.7 version is required. These are the algorithms used in the project, which is hierarchical clustering, first one. This algorithm groups similar objects, which is genes in this case, into clusters based on their similarity. It creates a tree-like structure called dendrogram, where each branch represents a cluster. Next one is k-means clustering. It is the algorithm that partitions the data into k predefined distinct clusters. Each data point, which is gene, is assigned to clusters whose centroid is closest to that data point. Next one is self-organizing map clustering. This SOM algorithm is a type of artificial neural network that employs unsupervised learning to produce a low-dimensional representation, usually two-dimensional, of the input data. Next one is principal component analysis. PCA is a feature selection algorithm that uses to reduce dimensionality of the dataset. It improves unrelated data and focuses on the most relevant and significant features, making it easier for the SOM algorithm to build clusters with pure and relevant genetic data. Coming to the methodology, we have designed these modules for execution. First one is to load genes dataset. In this step, the genes dataset is loaded into the computational environment. The dataset contains genetic data information for cyclooxygenase, which is COX1 and COX2 genes from various species. Next one is converting genes dataset to Excel format datasets such as CSV. To facilitate data manipulation and analysis, the genes dataset is converted into specific formats such as CSV, which is comma-separated values, which is widely used for data format. Converting the dataset into CSV allows easy handling of data in tabular form. Next one is gathering means of CSV dataset. In this step, the means of CSV dataset are computed. The means are likely calculated for each gene across different species. Next one is performing hierarchical clustering on the dataset. Hierarchical clustering is applied on the genes dataset. This algorithm genes with similar patterns or behaviors into clusters based on their pairwise similarity. Next one is performing k-means clustering. K-means clustering algorithm is used to partition the genes dataset into k predefined clusters. Each gene is assigned to one of the cluster based on similarity to the centroid of the cluster. Next one is performing PCA and SOM cluster data to get similar genes plotting. In this module, principal component analysis is applied as a feature selection algorithm to reduce dimensionality of the dataset. PCA removes unrelated or less relevant data, focuses on most significant components that contribute to the variance in the genes dataset. This step improves the accuracy of subsequent clustering. For execution of the project, first open the code folder. This is the code folder and this is the dataset folder in it. And on this dataset, we will be building the algorithms. These are the parameters of the project. This is the main code file, and this is the Python code file which is used in main code file while execution. This is the run.bat file. Instead of executing the project in command line interface, we have created Windows batch file for execution. So double click on it for execution. This is the graphical user interface of the project. With this, we can interact with the program, and this is created by using Tkinter library in the Python. First, click on the button Upload Genes Dataset to upload the data. Select the data and click on Open. Here, it is confirmed that data is loaded. Now, click on the button Convert Dataset to Excel, which will be converted into CSV format. Here, we have 1000 records and 11 columns in the dataset. Now, click on the button Run Hierarchical Clustering. Here we can see total 4 clusters are there, which are from 0 to 3. From the screen we can say that first record is in the cluster 3 and the second is also in 3, and it goes on in similar way. For each record we can see cluster label and all records in same cluster having similar genes. Here now I will be showing the dendrogram. This is the dendrogram. It is a tree diagram using a hierarchical clustering to show the similarity or dissimilarity between the data points or groups. It helps visualizing the hierarchical relationships and natural clusters within the data. The same color line represents the same cluster. So here the orange line is one cluster, green line is one cluster, red line is one cluster, and blue line is one cluster. I will be closing the dendrogram. Now I will click on Run Hierarchical Clustering. K-means clustering. Here we can see cluster labels for each record, which means first record from the dataset is 2. Similarly, it goes on. For each record, we got the cluster label. Now click on Execute SOM with PCA on genes data. Here, the SOM graph representing result of clustering genes using self-organizing map algorithm. The SOM graph is a two-dimensional representation of the genes dataset. Here, we can see where each gene is plotted in small circle on the map. Genes with similar patterns or behaviors are positioned or close to each other. Here, and the others are positioned in farther place. Now close this graph. Exit the GUI. Conclusion: researchers used cluster analysis technique on gene data to investigate the genetic algorithms of COX1 and COX2 genes from different species. The project created a phylogenetic tree that revealed separate branches containing genes from same species. Dendrogram clustering revealed insights into gene grouping. PCA and SOM were used to visualize genetic similarities in a low-dimensional space. This project demonstrates the ability of current bioinformatics in identifying genetic diversity and evolutionary relations, contribution to comparative genomics and evolutionary biology.
Thank you for watching video. For more projects, please visit our website www.trueprojects.in. For updates on latest project videos, please visit True Projects YouTube channel and subscribe.